these laboratory and field experiments allow us to watch evolution in progress. We can see the actual changes in the DNA between generations. So because we can study things for multiple generations, we can see how things change just so slightly over time. So here's one example. The grass on a golf course actually has 3 types. And I didn't know this because I don't spend a whole lot of time on golf courses, but if you do, then the grass that you have in the rough is going to grow pretty tall, and it's going to be pretty thick grass. And then the grass that grows in the fairway is going to be a little bit shorter, and the putting green a little bit shorter than that. And so you may think, well, yeah, they do that with lawnmowers. They actually, a single species of grass that's planted on a golf course, because on the putting greens they cut it so frequently, and on the fairway it's only cut occasionally, and then in the rough never cut at all, they've actually had this selective pressure that makes it, so here's our monthly cutting schedule. If you cut the putting green grass very, very often, then the grasses that sexually mature the slowest are going to be found in the rough, and those that produce the lowest amount of seeds. And then in the fairway, it comes to sexual maturation very slowly and produces a low amount of seeds. But if you're the grass on a putting green, then you have to mature very rapidly and have seed production that is very high, because otherwise when they come along and cut you down, if they cut it down and they can't mature fast enough and they don't produce any seeds, then they're not going to go on to produce any offspring, and they're not going to pass their genes along to those offspring. So in just a few years, the grass plants from one planted golf course actually evolve into 3 distinct populations, and that happens in a matter of years. So we've also seen this with antibiotic resistance. For those of you that have already been in lab this week, we talk a lot about penicillin and about Staphylococcus aureus. That's the bacteria that we were working with this week in lab. It is the bacteria that's found naturally on your skin. So if you were to take and just scratch your skin, you would have Staph aureus underneath your fingernails. And back in the 40s, they used to have it so that Staphylococcus would be killed off very quickly with penicillin. And then after the Staphylococcus aureus became resistant to penicillin, we found that very little of Staphylococcus aureus is killed by penicillin today. And the reason for that is, it used to be penicillin would just destroy all of those bacteria, but then as people stopped taking the full rounds of antibiotics and humans took so much penicillin, it got to be that we needed more and different types of antibiotics in order to kill off things that used to be able to be very easily killed. And that's why we're coming up with antibiotic-resistant bacteria that are very, very dangerous in our hospital systems. And for any of you that are in athletics, that's very dangerous for athletes, because a lot of times it's found with people that have very close contact. So when we talked about our fruit fly experiment, where we wondered, can fruit flies evolve so that they can resist starvation longer, we found that after many generations, in this case it was 60 generations, that with natural selection taking place, the population changes. And so the fruit flies that we have at the end of the experiment are very different than the fruit flies we had at the beginning of the experiment. The grass in the golf course is very different at the end of the experiment than they had at the beginning of the experiment. And the antibiotic resistant, the staph that they had at the beginning, is very different than the staph that we have today. And so we've seen lots of different populations that have evolved, some over long periods of time and some over very short periods of time.